late. Yesterday you have learned about that how proteins can be extracted from the sample. It could be microbial cell or animal or plant cell. Though there will be variation depending on how hard your cell wall is there, different methods of extraction will be used. But yes, you have to make a crude extract. Then you fractionate. Fractionation is done by using different methods, and one of the most common method is a salting out. And then we have talked about the dialysis, the need to remove the salt from the solution. And then we have talked about the column chromatography. In column chromatography, we have talked about how we can vary the protein charge, and based on the protein charge, you can separate the protein molecules. Then you can also separate them based on the size. So these two portions we have already covered. And you have seen that if there is a variation in a protein charge, what you can do is that you can use ion exchange chromatography. So as you have seen in ion exchange chromatography, the major, important thing is that what kind of exchanger you are using. So depending on the type of a protein, the nature of a protein in terms of their charge, you can select your resin or a polymer, which can be made as a matrix, and then you can separate these proteins. Then we have also talked about the importance of a column length to get better separation. Uh, and then we have moved on to the gel filtration chromatography. Now, in gel filtration chromatography, or what you have learned is that, so this is the one which is an exchange chromatography, where you are using resin which has a charge on the surface, and that's why charged protein binds. But on the other hand, you have also learned that we can have resin which has a pore, and because of the pore, proteins which are of small size that can enter inside the pore, and they can be eluted from the column at a later stage, compared to the one which are bigger protein which has a bigger molecular mass, and they are residing outside these beads. So this part we have already covered. Now what we are going to talk about is that proteins or other molecules, they can be separated on the basis of their affinity chromatography. Affinity, as the name indicates, it is based on the binding affinity of the gel. So whatever the gel we fix, so the column remains same. You have the same glass column or plastic column, and what you do is that you just vary the matrix, the gel which you are using. In first case, you have used gel which was a charge. In second case, where you have used gel and this gel was having pores, so you have used two different types of gel. Here, third property of the gel is important, where what you do is that this gel has got beads, and these beads, they are covalently attached with a chemical group, and this chemical group is called ligand. So you can see there is a bead, there is a center, you have a bead, it has got a ligand, and this ligand will have affinity for a particular type of protein. So to explain it better, let me just try to give you some example. Suppose I'm interested in a separation uh, of an enzyme. What I will do is that I will take something which can bind with enzyme, because enzymes have a binding site. So it is very easy to select a ligand, because anything which has a structure like substrate, they can bind with the enzyme active site. So what you do is that you can make ligand which has capability to bind with the protein. Now in this case, what will happen if I take a protein mixture which is added in the column, the one which has affinity for the ligand, they will bind, and rest protein, they will come out of the column. So what you do is that you keep on pumping buffer solution, and thereby the one which is having a specific affinity with the ligand, they will bind with the column matrix. Other proteins, they will be coming out of the column first. Then the question comes that, okay, what I have done is that we have allowed the ligand and protein to have interaction, and thereby the protein is a binding here. How we can separate them? The separation can be either using the salt concentration, or you can use free ligand. For example, I have also given example that you have known about the lectin. What lectin does? Lectin binds with specific glucose molecule or specific carbohydrate molecule. So if I choose a carbohydrate as a ligand, what will happen? The lectin will bind here and the rest thing will be removed from the column. Now what you do is that in the next step, either you change the salt concentration and thereby lower the interaction of the ligand with the beads, or alternatively you can use free ligand. Now here you have immobilized the ligand. Now if I use free ligand, they can also replace the one which is, so what you are doing is that you are trying to reduce or weak the interaction, and thereby you can separate your protein molecule. So why salt allows this protein to come out? Because salt weakens the binding of protein to immobilize ligand, and that's why because it is interfering. If the interaction is by ionic interaction, as you know, the salt can interfere because salt are a smaller molecule. They will bind more in numbers, and thereby they will replace the protein molecule. Same way, you can use free ligand, which can compete, and after competing with the matrix, they will allow the protein to be liberated. So this way you can use affinity chromatography. And so let's talk about how these three types of chromatography differs from each other. First, here we have shown that how ion exchange chromatography is important. So if I have a protein which is different in the charge, so if I know that my protein of interest is a charged one, then I will use the technique called as ion exchange chromatography. There is another technique which is gel filtration. So if I know that, okay, I have some contamination in the protein, which is having difference in their molecular weight. So what we'll do is that we can use gel filtration chromatography or size exclusion chromatography. On the other hand, at the last step, if I know that, okay, my protein has been purified, but it is not completely purified, then we can use affinity chromatography. I will also talk about, is it possible that we can have a combination of all the technique, or is it the single chromatography will give you the all kinds of separation? Answer is that what you require is, many times you require the combination of all types of chromatographic experiment to purify protein. Now, another question comes that if I'm purifying protein, how I will get to know that proteins are being purified? So I'll also talk about how you test after every step of purification that your protein is getting purified or still it remains same. Now, proteins, once you purify them, there is one of the way by which you can characterize it is known as electrophoresis. So, electrophoresis is a very important technique for separation of protein. Um, and what happens that this method generally, because you are passing the electrical current, what is happening that it can affect the structure and the function of the protein, but it is a very strong analytical tool. And that's why most of the time, whenever you do your experiment, if you are working with a protein, you will definitely use electrophoresis. In fact, in your experimental part, you will be doing this electrophoresis where you will be able to separate protein based on the electrophoretic technique. Of course, the technique here, when we are using electrophoresis, it is assuming that the proteins, they are charged molecules, and because of the charge, they will be separated if we subject the electrical field. The advantage of this technique is that while you are separating it, very quickly you can visualize that they are getting separated. And one can estimate it quickly, and you can also find out how many number of proteins you are present in your mixture and what is the degree of impurity. I will show you one of the electrophoretic separation which will show you how the proteins can be separated and how the different techniques are being used to, separate the protein in a larger scale and then check it whether they have been purified or not. Then electrophoresis can be used to determine crucial property of the protein, such as isoelectric point. So now question is, what is isoelectric point? Isoelectric point is a protein in a protein is given by the point or a pH at which the net charge on the protein is zero. So what it means is that protein, they do have a charge, but if I keep on changing the pH, they can change their net charge. Is it clear to you? Have you understood the concept of isoelectric point?
Yes, sir.
Okay. So because, suppose I change the pH, what I'm doing is that if you recall, we have talked about the amino acids. Amino acids are amphoteric in nature. That means they have both positive charge as well as negative charge. Now, depending on what pH you have, the net charge of the protein will change because their individual amino acid, they will have certain charge, and that charge will be reflected based on what pH you are using. So let us talk about some basic concept of electrophoresis. In electrophoresis of protein is generally carried out in a gel, which is made up of cross-linked polymer, which is known as a polyacrylamide. So most of the time, what you do is that you see that polyacrylamide gels, they are being made in the lab itself. So you will be able to prepare these gels. You can cast the gel, and then on this polymer, what you will see is that you can separate your protein. Why people they use polymer, this polyacrylamide? Answer is that these polyacrylamide gel, they act as a molecular sieve. So what it does is that they can separate protein based on their size as well. So the protein will not just flow just because of the electric field. They will also be restricted to the molecular sieve which is present because of the cross-linking in polymer, which is a polyacrylamide. In fact, polyacrylamide is one of the most common protein which is for used for protein separation. You can also use this electrophoresis for separation of DNA. So in your experiment, you will be separating both DNA as well as the protein using polyacrylamide gel for protein and agarose gel for DNA. So polyacrylamide gel is the one which on one hand acts as a molecular sieve, and that's why they slow the migration of protein approximately in the proportion charge to mass ratio. So depending on the mass, you will get that they are going to be, slowing the migration of a protein. Now, how this electrophoretic mobility is being determined? As you know, it is a very simple that the electrophoretic mobility mu can be given by a molecule in a ratio of velocity versus electrical potential. Now, this also is very simple thing that if something is moving in an electric field, what are the other factors going to determine it? As you know, that there is a one thing which is important. Of course, if you increase the electrical field, the mobility is more. But at the same time, it also depends on frictional coefficient. So if I allow a molecule to move in different matrix, their because of the frictional coefficient, their movement will be different. And what else can affect the mobility? Another factor which can affect the mobility is the shape of the protein. If it is a rod shape, it is a circular, it is a triangular. So depending on the shape, they will also determine, because that is going to determine how the protein is going to move in the electrical field. So the migration of a protein in a gel during electrophoresis is function of its size and shape. The electrophoretic method commonly employed for the estimation of a purity and molecular weight makes use of a detergent. So here we use a detergent. Why do we use detergent? The reason here is that different proteins, they will have a different charge. So if they are having different charge, actually they should move based on their charge. But what has been done here is that a detergent, which is a sodium dodecyl sulfate. Most of you have known about this. This is a very common detergent. The structure is given here. This is your, there are 12, this carbon chain, and then you have a sulfate, and of course at the terminal you have a sodium bond. So what happens when we treat the protein with sodium dodecyl sulfate? It binds with the protein, and each protein, suppose I have one molecule of protein, there is a 1.4 molecules of SDS which is going to bind. And because of this binding, what will happen? The net negative charge on the protein surface will be very high. So because of the large net negative charge, the charge which is because of the protein, which is intrinsic charge, that will not be counted. So that becomes, a negligible factor. And that is why when protein moves in electrophoresis, it is because of the charge to mass ratio and not because of the intrinsic charge of the protein. So when you get the separation in polyacrylamide gel electrophoresis, it is because of the molecular weight. And that is why you will learn that this method can also be used for determining the molecular weight of the protein. Now, once you're separating the protein, proteins are colorless. How we get to know that they got separated? So that's why there is a reagent known as a Comasi blue, which binds with the protein but not with the gel. So what you will see is that wherever the separation of the protein is coming, they are getting, colored, whereas the other part of the gel remains uncolored. Now, this is a typical electrophoretic system which has been shown to you. Here you have a negative and here is a positive terminal, and that you can connect with the power pack. And this is the gel which has been made. So this is the gel which you have made. And what you do is that you use a comb. And of course, this part you will be doing and learning experimentally, where you will learn that you can create wells. So here you see there are different wells. So what you do is that if I put a template kind of a thing or a comb kind of thing, so each part, the gel is not polymerized. So while polymerizing, you put a comb, and because of that, you create these gels. So once you remove the comb, this place is working like a well. And each well, you can put the protein. So normally, you put something like 10 microgram protein, which can be separated when you run the gel electrophoresis during your experiment. So here, what you do is that you load different samples, and as I have shown you in the previous slides, that you can separate the protein based on different techniques. So suppose I first take a protein and separate them. Suppose I have separated them by first by precipitation method, where we have used ammonium sulfate, and then we take a, we analyze how many bands you are getting. Then I run the ion exchange chromatography, see how many bands we have got. Then we run with another technique, affinity chromatography, and see how many bands we are getting. So every time you will see that there is a certain level of purification which you are achieving. Is it clear to you?
Yes, sir.
So, because ultimately we are going to use this technique for determination of the molecular weight, what we do is that we also use a standard marker. So on the right hand side, the first lane, you will find that there is a certain number which is given, say 97,400, 66,200. So this gives you molecular weight of a protein. So these are the protein mixture which has got known molecular weight, and that's why we know that the top one, the one which is having highest molecular weight, is sticking over here, because that will have maximum restriction to flow in the gel. Whereas the smallest molecular weight protein will be going in the bond, in the lowest part. Of course, experimentally, it is also possible that if I run the gel, it can run out of the overall, this whole area. So what is done is that very small molecular dye is put. So at the end, what you find is that, for example, this one, the terminal one, which is not 14,400, below that, this is known as a tracking dye. So you can track the movement of this, though you will not be able to see these bands when they are getting separated, but you can see the tracking die. And after you run the gel, what you do is that you stain them, de-stain them, and this is the complete procedure by which the dye will bind specifically to the protein, and then you can separate them out or you can visualize them. It's a very simple technique, and that is why it is very popular. Now what we are going to talk about is here the gel which has been given. Of course, this is done in when you do protein expression. It's also very important tool for molecular biologists to see that whatever cloning of the gene they have done, they are able to see the expression of the protein, because ultimately aim is to look at they are getting the protein expressed and purified. Here what I'm showing is that the first lane is showing the marker. Uninduced cell, induced cell, right now I will not confuse you because these are the one where we are looking at the expression of the protein. In a cloned, if I take a gene which is going to be expressed, here we are looking at the, separation or the amount of protein of your interest, how much they are going to be expressed. Now this one becomes important for you. The fourth lane, which is showing soluble crude extract. This soluble crude extract is the one which you have isolated for protein purification from, say, microbial cell. And then you can see ammonium sulfate precipitation, anion exchange chromatography, cation exchange chromatography, purified sample. So what you find is that the purified protein will have a single band. Of course, it is not showing many bands. For example, ammonium sulfate precipitation, you can see more than one band. Same way, there are bands, but you will find very faint bands for after anion exchange chromatography or cation exchange chromatography. And in case of purified protein, you will not see any other band. So now we are going to extend the same chromatography to learn how we can use sodium dodecyl sulfate polyacrylamide gel electrophoresis for determining the molecular weight. I have also talked about that there is another technique where you can determine the molecular weight, which is the gel filtration technique. Here, for example, you see there are two lanes which I have given. One is the one where we know the marker, where we have a standard protein. So there are different proteins. So this is given as their molecular weight. So there are molecular weight based on which they have got separated. And this is the one which is unknown protein. So if I have to determine the molecular weight of this unknown protein, what I'll do is that I will make a plot between the log of molecular weight of all these proteins. So this is the log of molecular weight of the protein, and this is a relative migration. So what is the relative migration? Suppose you have put this spot here and this protein has moved up to here. So the center of this spot will be taken as the relative migration of the protein. So for myosin, this is the relative migration. For example, this unknown protein, the relative migration is up to here from this spotting where you have done the spot. So you can find out you can plot between log of molecular weight versus relative migration. So you have done this standard, you can do so. And then for unknown protein, now unknown protein will come somewhere based on that, their relative migration, you can find out the log of molecular weight of that protein. Is it understandable? Have you understood this concept? Hello?
Yes, sir.
Any doubt? If you have any doubt, please ask. So it's a very simple technique. You can do in your lab. You can demonstrate this. In fact, you will be doing this part of experiment in your lab. Now I'm going to move on to a technique which is similar to electrophoresis, but it has one more step involved in the electrophoresis. What you do is that you can determine the something which is very, very important, where one can separate not only based on their charges to mass ratio, you can also separate them based on their charge. And this technique can even separate if there is a difference of one charge on the surface of the protein. And this technique is known as isoelectric focusing. So what does it mean? It means that it used to determine, first thing is that it is used to determine the isoelectric point of a protein, which I have already defined, that isoelectric point is a point at which net charge on the protein surface is zero. Though there will be charge, there will be positive, equal number of positive charge, equal number of negative charge, and that's why the net charge is zero. So here what we do is that we make the gel. So this is a gel strip, and this gel strip is immobilized with immobilized pH gradient. So you have a gradient of pH, and this gradient of pH is in 0.1 increment. So what you have is that, for example, it is from pH 9 to pH 3. You will find that there is a decrease. You have several pH present on this strip. On this strip, what I have done is that I have taken the material. It is basically a zooter ionic material which is present, and they are from different pH. So you will find there is some pH here, some pH here. So this pH you will find across the strip. And what you do is that you apply your protein sample, and then you apply the electrical field. And after applying the electrical field, you will see that the protein will start migrating, and they will stop migrating at a point where their pH is equal to isoelectric pH. And that is what is determined as a isoelectric point. So a pH gradient you have established, which allows the mixture of the low molecular weight organic acids and bases to be distributed. So the one which is creating the gradient is basically low molecular weight organic acids and bases. And what you have done is that you have applied the electrical field, and then you applied the protein mixture as you have seen here, and then you get the separation. So this is another technique by which either you can separate the protein at a very, very low quantity, because here it is extremely uh, sensitive method. So very, very small quantity of protein is being purified by this technique, and you can separate the protein which is not separated otherwise using molecular weight. Now, once you have separated and purified the protein, you need to know how much protein is present. To purify a protein, it is always essential to have a way of detecting and quantifying that protein in the presence of many other proteins at each step of procedure. So I'm going to talk about, suppose you have taken a crude sample, what should be the way to estimate how much protein is there? So there are techniques. I have not discussed about that, but yes, one point you already know, that if I have to estimate a protein, what can I do? The simplest way is that I can take the spectrophotometric reading of the protein, which is based on the absorption, and the absorption value will reflect that how much is the concentration. So where do we see the absorbance of protein? At what spectrum or what wavelength we measure the protein?
280 nanometer.
Yeah, so at 280 nanometer, because it is assumed that most of the protein, they will be having either tryptophan or tyrosine, which absorbs at 280 nanometer. So if I take the absorbance at 280 nanometer, we can estimate the concentration of the protein. So far, we have not talked about the concept of estimation of a protein. We have not talked about how you can, take the extinction coefficient or the absorbance of the protein. But yes, it can be done. There is another method which is based on the binding of a dye with the protein. So depending on the concentration of the protein, the dye binds with the protein surface, and you can take the reading. So there are different methods. You will be doing at least 2 or 3 methods of estimating protein, because it's very important whenever you do, your molecular biology and you are expressing your protein, your concentration of a protein is very important. Even when you are purifying, it is very important that how much protein you have purified. So one thing is that we are looking at the protein purification. The other thing is that out of that protein, which type of protein we are going to estimate. So suppose I have 6 protein or 10 protein which is present in our crude extract, or even after purifying by your or separating it by the salt precipitation, we should know the protein of our interest. And to take a best example, if I have several protein and I'm looking for a particular enzyme, I should be interested in that how much that enzyme is present. So to take a very clear example, if I want to measure that I have taken an extract and how much protease enzyme is present or how much lipase enzyme is present. So we should know the procedure to estimate the enzyme. And to some extent, we have talked about how you can estimate enzymes. So actually, when you extract these proteins, or you know the difference between proteins and enzymes, proteins and enzymes are similar in terms of the structure in most of the cases, except that enzymes have binding site. And because it has a binding site, you can estimate, you can measure them. So because enzymes, they have catalytic power, you can measure them. So for this purpose, what we need, so you should need the overall equation. Suppose I have to measure the protease, what I need to know is that how much is which substrate it is going to use and what is the product of the reaction. So suppose I am looking at the protease, I should have a protein, I can use the enzyme protease, and what you get is that you get hydrolyzed protein, that means different amino acids. uh, you should also have analytical procedure for determining the disappearance of the substrate or appearance of reaction product. This is very simple. You have learned that in case of enzyme, if I want to measure what is the activity of enzyme, you should always know that either you should know that how much your substrate is being consumed, or you should know how much your reaction product is being formed. Then it also depends on how much cofactor is involved, because many enzymes, they may not act if the cofactor is not present. And that's why it is important that we should know whether the enzyme is requiring cofactor or not. Uh, then we also need to know what are the optimum concentration, optimum pH, at what temperature, because suppose I'm using at say 60 degree, what will happen? All the enzyme will get denatured and I will not be getting any activity. Suppose I use at pH 12, enzyme is getting denatured, I will not get any activity. Suppose I'm using in a concentration range which is not optimal concentration, you will not get you get all of the enzyme getting saturated. So depending on all these conditions, you can measure the activity of an enzyme. Now let us see how or where it is advantage. So normally enzymes, they're used, whatever we use at their optimal pH, and pH is normally around 7 pH, because if you see most of the organism, their pH is 7.4 inside the cell. The convenient temperature to be used is between 25 to 38. 25 is normally considered as a room temperature, and 37 degrees is considered as a body temperature. So in one of the temperature, most of the enzymatic assay is done. And also very high substrate concentrations are generally used. So what you are looking at is that all the enzyme active site, they are saturated, so that you can measure the initial rate of the reaction experimentally. And here I would like to define what is the enzyme activity. So enzyme activity, as per the international agreement, as I told you earlier that there is an enzyme commission and that determines like IUPAC, it also has certain guidelines. And by that guideline, 1 unit of enzyme activity for most of the enzymes is defined as the amount of enzyme causing transformation of 1 micromole of substrate to product per minute at 25 degrees. So you have defined the optimal constant condition. You have defined that you are looking at the transformation of 1 micromole of the substrate to product, and you have also defined the time. So this gives you international unit of enzymatic activity. And then one can also determine the specific activity. The specific activity is given by um, is the number of enzyme units per milligram of the total protein. So suppose you have purified enzyme and you are getting activity, it does not mean it is the same specific activity. I will try to give an analogy and explain it, what is the specific activity or activity. So after each purification step, suppose you are running gel, you do the enzymatic activity and you can get to know how much protein is present. So you can determine two things you have to determine here. One is the how much is the protein concentration and how much is the activity. So once you know these two, based on the ratio, you can determine the specific activity. I'll give you a chart and show you that how these two differ in terms of their properties. So activity decreases most of the time when we go from one step of purification to another step of purification. Activity will keep on generally decreasing. But what happens, the activity decreases because there is always some loss during the process, either due to inactivation or non-ideal interaction with chromatographic material. For example, you are using, say, ion exchange chromatography. Some of the protein, they are not really binding with the gel matrix. So what will happen? They will pass out. And because of that, the actual protein which you are interested in separation, they are not completely separated. It will also depend on how much protein we are loading in the matrix. If the protein concentration is very high, all the binding has occurred, they will not bind, they will be passing out of the column. So because of these reasons, because you may not have the optimal condition, the binding may not be 100%. So you will be having always loss of the protein during the purification step, and that is why the activity will keep on decreasing. But and the total protein also decreases. So from one step to another step, you are losing protein as well as protein of your interest. But what you want is that in a successful step, the loss of non-specific protein is much greater than the loss of the activity. So what you need is that even if you are losing protein, you should lose a protein which is not of your interest. And that is the way your specific activity should increase. So during a process of purification, the activity decreases, constant that amount of protein decreases, but specific activity increases. Okay, here I have given you that if I'm looking at the transport protein, we can go for their transport property. If I'm having hormones, you can look at the biological effect. So there are different types of proteins and they can be measured by different means. Then we are, this is the table which is going to explain how the purification steps looks like. And we have taken a hypothetical enzyme. Just read this table. Here we have taken crude extract and the volume is quite large. So you have a huge amount of the protein which you have extracted from the cell. The total protein is say 10,000 and the activity based on how many microgram of protein has been converted into the product per minute. It is something like, this is the unit which is given. So it is 100,000 units which you have. And specific activity, so when I divide activity by the total protein, the specific activity comes to 10 units per mg. So when you are looking at activity, it is in units. When we are looking at a specific activity, that means unit per milligram of protein. Then what happens, we have precipitated the protein. So you have got precipitate, and this precipitate, the volume goes down because you precipitate and then solubilize and then start estimating. Here, if you look at the protein, substantially you have lost protein. But what should be the purpose? Purpose should be that though we lose the protein, but the activity should not be lost. And how you can do is that you can optimize that how much percent ammonium sulfate you are using. So at different percent, you will find that you will have different recovery of a protein and their activity. Next is ion exchange chromatography. From this step to this step, when we are moving, you will again see the loss of the total so you are reducing the volume as well as the protein concentrate. Protein is also being reduced. So here, what you are looking at is continuously you are reducing the volume as well as you are reducing the protein concentration, as well as your activity is reducing. But what you are finding is that your specific activity keeps on increasing. Each step you are finding your specific activity is increasing. That means in that fraction, your protein of interest is increasing. So affinity chromatography, as you can see, is used as the last step, because otherwise what will happen, affinity column will be blocked if you use in the first step. So unless proteins are substantially purified, we don't use affinity chromatography. Now, this order of purification step may be different. It could be, but most of the time, you have to have crude cellular extract unless proteins are excreted outside the cell. Next step is mostly ammonium sulfate because it is, separating in the simple procedure. Then you go for ion exchange chromatography or gel exclusion chromatography. Any one of them can be used, and you can determine the order depending on how much protein you can retain and how much activity you can find. Now, this I'm going to give you a very simple example of explaining how activity versus specific activity can be explained. So you can see there is a difference between these terms, and there are two flasks, flask number 1 and flask number 2. Both of them you will find that they are containing marbles. And even if I say the flask 1 and flask 2, both of them, they have different colors of the marble. And what you find is that if I look at the number of red marble here, U1, 2, 3, 4, 5, 6, whatever is the number, even if the same number is present in the second flask, their other proteins, they have been separated out. That's why if you look at the title, we are saying that unseparated proteins can be quantified. So what you are looking at is you are looking at how much protein you can specifically purify of your interest. So suppose I say that each marble is equivalent to your type of a protein, one type of protein. So what we find is that here the protein concentration is high, here your total activity is high, but at the same time the activity, because of this red marble, is not in terms of a fraction is that much high. So what a specific activity does is that a specific activity is specifically present the concentration of a protein of your interest. So in summary, we have talked about the proteins can be separated and purified on the basis of difference in their properties. Proteins can be selectively precipitated by change in pH, temperature, and particularly by addition of certain salts. Now, when I say temperature and pH, of course, protein, if I change the pH, some of the proteins, they can be precipitated, as you have seen in case of, different ways in which your charge, if there is no charge, protein will be precipitated. This experiment can be demonstrated very simply by changing the pH. Suppose I take a milk, and most of you have seen milk. If I lower the pH of milk to say 4.1 or 4.2, the protein gets precipitated. Why? The protein has, protein which is present in the milk, which is a casein protein, that gets precipitated. Why? Because the proteins, they will not be able to, be solubilized. They will be separated out from being soluble in milk. So that's where you can separate the protein from milk. The same way can be used for separation, but most of the time what we do is that we use simple method where we use salt, because pH can also change the activity of enzyme, which may not be recovered at the later stage. So the most preferred method is using salt. And which salt? Ammonium sulfate. So you can use wide range of chromatographic techniques like based on their size, binding affinity, charge, and other properties. These include ion exchange chromatography, size exclusion chromatography, affinity chromatography. I have not discussed about high performance liquid chromatography. It is based on different method. High performance means this is also known as earlier known as hyper pressure chromatography. So you will learn the concept that if I put pressure through the peristatic pump, which is delivering the your buffer, that can improve the separation. So this is a very sophisticated technique for separating the proteins. Electrophoresis can be used for separating the protein on the basis of mass or charge, and SDS electrophoresis and isoelectric focusing are the one which are very important for protein separation. And not only protein separation, you can keep on getting the exact knowledge whether you are getting your protein separated or not. So, and all the procedure requires method for quantifying. So you have learned about how enzyme can be assayed and quantified. And with this, I'm through with this class. So if you have any quick question, we can discuss. Any question from your side? Hello?
No, sir.
So if there are no more questions, I will stop at this point. Thank you very much.
Thank you, sir.
Thank you, sir.